name is Cedric Espenel. I work at the cell science imaging facility at Stanford. And so first I wanted to give a few words about where I work, the cell science imaging facility. So it's a Beckman Center supported university service. So we provide help. So we have light and electron microscopy technology over there. We have some regular light microscope, such as confocal wide field microscope, but we also have more advanced microscopy technique, such as the lattice light sheet. So you have here a little bit of the light sheet, but it's similar technique. We have multiplexing technique. We can do super resolution also. And we have some electronic microscope. And if you're interested, you can go to our website to see the type of technique we have. So we are a service center, so we take care of the microscope. We also provide consultation, advice, training, and education on how to acquire images and how to use a microscope. And more recently, we have started to do some more image analysis and help with image analysis, which is something that was lacking at Stanford. And actually, I was at the ImageXD workshop in 2016, where I learned how to use Python to do the image analysis. And since then, I've been doing a few projects. Some have been published, actually recently. And we have a GitHub GitHub page where you can go and see some of the projects we have been working on. So the project I'm going to present today has been done in collaboration with the Anne Villeneuve lab at Stanford, more specifically with Chloé Girard, a postdoc in the lab. And in the Anne Villeneuve's lab, they're interested in meiosis. So the meiosis, I'm not going to go too much into the biology, first of all, because it's not really my domain, meiosis, but if you have any question, I'm happy to answer. But the idea of meiosis is to go from a diploid cell, so where you have two sets of chromosomes, to a gamete where you have only one set of chromosome. And during that process, you have something called homologous chromosome recombination. And you have actually the chromosome, they kind of cross over each other. So that's what you can see in the image on the left. And the formation of this crossover imply recruitment of a lot of different proteins. And the Anne Villeneuve lab are interested in the timing of the formation of this crossover when they happen during meiosis, which protein are involved in that crossover, and when does it happen. And so they do their study in C. elegans. So C. elegans is a small worm, which is very convenient to work under a microscope, mainly because it's transparent, so you can see through it. And also it's easy to fix it onto a gel and make it stuck there. And also, you can also follow the whole process of meiosis inside the worm. So here you have an image where on the top you can see where the gonadal arm is where the meiosis happen. And at the bottom right, you have an image where you can see the progression from meiotic region to the different stage of the meiosis. And the Pachiten region, it's kind of hard to see, but it's where this crossover happened. It's the main region of interest for their lab. So if you look at the C. elegans under a fluorescence microscope, that's kind of how it looks like. So here in blue, you have the chromosome that are labeled. And this is a maximum intensity projection. It's a stitched image. It's a volumetric image, and you can see from the different stage of the meiosis in that image. And so the region of interest for the Anne Villeneuve's lab was a lot the Pachiten region. So as I say, we are a service center, so we provide help. And so Chloé Girard came to us in 2018, and they wanted help with their image analysis. And so their main issue was that they were doing everything manually. So they will go into ImageJ and find the nucleus, so this little circle, and then count the number of green dots there is on the nucleus. And they wanted to have a more automated way to do it, to spend less time in front of the computer, basically. And also being able to be more reproducible and having different people getting the same results. So the design specification of the pipeline was to first find the position of each nucleus, then being able to extract the distance of the nucleus from the tip of the gonad, because they wanted to know in which stage of the meiosis they were, and then finally count the number of foci or crossover. These dots are the crossover onto the chromosome, between the chromosome that you see in green, and be able to count these dots and the number of these dots per chromosome. So, as a reminder, this image are so the image they work with are stitch image of 512 by 512 image. You can have 15 to 30 of this image, and they are Z-stack. So there are a lot of dimension to this image, and you also have up to 4 channels. So 1 channel for the chromosome and different color for different proteins that are involved in crossover. So for my part, what I did is I didn't work directly on the stitch image. They were a little too big for me to deal with. So I work in the individual image, and then I go back to the stitch image afterward. So the first challenge was to find the nucleus in the image, mainly because of the limit of resolution of the microscope. This chromosome overlap. Also, so you're in volume, but all the chromosomes kind of overlap and touch each other. So being able to find where they are was the first challenge I had. So to overcome, and again, I work more at the basic level. I don't use any deep learning, but I use the Dalal and Triggs method to be able to find this chromosome. So that's a method that was used originally to find human in a picture. So I adapted to my project to be able to find the nucleus in my images. So I created a training set using ImageXD, and I just look at the nucleus in focus. So I create a training set, then I convert the image into a histogram of oriented gradient using scikit image, a function in scikit image. And then I use the feature from the hop descriptor to train a classifier using a linear support vector machine. And I get my classifier, then I slide a window over the image and over the volume of the image. And for every window, I will find the hawk feature for every window. And then, so then I check if there is a nucleus at every window. So out of that, I get a heat map that will represent the probability of having a nucleus at a specific region. And so just to remind you that it's this is a volume volumetric heat map. And this actually was done using this little video was done using Napari that I am using a lot these days, and it's been extremely helpful. So then, I can find all the different nucleus in the volume. The next step was to find the next step was to find the crossover in the image. So for that, I just use, again, a scikit image function. I use difference of Gaussian to find the foci. And this is actually also a little video that I made using Napari, where you can see how I highlight the foci in the image. Then I try to clean the image to remove the non-specific foci. I create a binary image of the nucleus of the chromosome, and then I can remove all the foci that were, all the dots that were found outside the nucleus, and I get to something like that, where I have, and then the next step is I actually use the result from the machine learning, where I create a volume around where I found the nucleus, and I count the number of foci there is inside that volume. So relatively straightforward, and I get the number of foci there is per nucleus this way. And I was able to compare with what they were getting manually, so I get a lot more nucleus, but overall I get about the same number of foci that they were getting what they were counting by hand. Finally, the last step was to figure out the distance of this chromosome to the tip of the gonad. And for that, I actually just use, the first step is actually use the metadata that are saved with the file I'm using. So I use the stage position for the different tile image. Then I can measure the accumulated distance, and I create this kind of skeleton of the gonad. From there, I can just plot back the result of the nucleus that I found using the train classifier, and then I can get kind of a heat map of plotting the distance from the tip. So for that, I convert the skeleton that I had generated using the stage position into a two-dimensional tree, and then I just query the nucleus that are closest to the query point. And finally, I can plot the number of foci versus the distance from the tip of the gonad, and I get this type of result where so here is plotted the number of foci per nucleus, and you can clearly see that in that region you have almost no foci, and that you get to this Pachiten region where you have formation of crossover and get an average of around 6 foci per nucleus, which is expected for what we are using. And yeah, that was about it for that project. So I wanted to acknowledge John Mulholland, who is the director of the facility, who found money to develop this project, because we actually do it freely. So the Beckman Center is paying for it, and also very open to me doing all these things. I also want to acknowledge all the different, so I said that I was at the ImageXD in 2016, but also I've been using all these different blog and tutorial that are available online, and have been extremely helpful for me to be able to work with microscopy images. Yeah, and also the different packages I've been using, so you're probably familiar with all of them. I will just mention briefly Python bioformat, which, because I work in an imaging facility, there is different microscope, different company that have all their own priority format, and it's this type of file are kind of complicated to work with, and fortunately there's some people that deal with that, and I'm able to use their work. And with Python bioformat, you can get out the metadata of your file, but also an image as an NumPy array. And also, I wanted, you have heard about an array before. I just wanted to mention that it's extremely helpful, because I've been struggling for a few years to find a good way to visualize this data, microscopy data that can be have a lot of different dimension to it, and it's just been very helpful to work with an array in the last months, I guess. And yeah, that's it for me. Thank you.
I have a question. That skeleton, it looked really hard to detect that. Did you use metadata, or was it detected just from the image?
Oh, yeah, that guy. That's just the metadata. So this one is a stitch image. So you have you have all these images that are kind of like that. And when you use the metadata, so using Python bioformat, you can get actually the stage position. So the microscope move, and you can get back the stage position from your metadata. And so all these orange dots here is actually the stage position of the image.
Okay.
And so then you can just get back the skeleton of the stitched image this way, which is easier than trying to figure out the actual skeleton of the stitched image. Yeah.
Questions? All right. Well, thank you very much.
Thank you.